Thrombosis occurs when blood clots block veins and arteries. Symptoms can include pain and swelling in one leg, chest pain or numbness on one side of the body. Complications of thrombosis can be life-threatening, such as a stroke or a heart attack. The successful nutritional approach to reduce thrombosis risk doesn't depend solely on a set of antithrombotic nutrients, instead supporting a multifactorial approach that includes nutrition and lifestyle interventions can reduce the risk of thrombosis. These include abnormal blood lipids, chronic inflammation, hypertension, elevated plasma homocysteine, and obesity. Platelet activation and aggregation occurs via a complex multifactorial process. Several natural ingredients can target the varying steps that are involved in clot formation, and a diversified regime can provide multiple defences against the aberrant clotting. Olives have a history of use against high blood pressure, atherosclerosis, and diabetes. The leaves contain the active iridoid compound oleuperin and oleacin, and these are thought to be responsible for its blood pressure lowering and cholesterol lowering properties, and this was demonstrated in recent human trials. In lab tests, olive leaf extract also demonstrated antiplatelet activity in blood isolated from healthy male volunteers. The high oleuperin extract from the olive tree also inhibits the aggregation of the human platelets in lab tests, especially for those with type 2 diabetes. Hydroxytyrosol and hydroxytyrol acetate are two metabolites of the oleuperin, and these are found in the olive fruit and the oil, and they have a well-established anti-inflammatory and antiplatelet activity in lab tests and in animal models. Phenolic-rich olive oil preparations have demonstrated decreases in the production of pro-inflammatory and pro-thrombotic factors in human studies. The hydroxytyrosol acetate inhibits platelet aggregation with an efficacy similar to aspirin in vitro using whole blood samples from healthy volunteers. Hydroxytyrosol-rich extracts at 25 mg a day for 4 days reduce the production of the pro-thrombotic factor thromboxane in a pilot trial on 5 diabetic adults. High-fat diets rich in olive oil lowered the plasma levels of several clotting factors in a large study on 20 healthy young volunteers. Tea consumption has established protective effects on cardiovascular health. Reductions in the risk of coronary heart disease and stroke from the tea consumption have been confirmed through the analysis of several population studies. Purified green tea polyphenols, such as the EG-CG catechin, increase the clotting time in rats and reduces platelet aggregation in isolated human platelets. Human trials of tea consumption and thrombosis risk have had mixed results. While short-term consumption, 2 weeks of the green tea, showed no measurable effect on the platelet activity, long-term studies showed modest improvements in the platelet function. The most promising results have been observed in a randomized, blinded trial of tea consumption. 6 weeks of black tea consumption at 4 cups a day in 37 healthy volunteers significantly reduced the platelet activation, as was measured by the presence of platelet aggregates. The tea catechins and the flavonoid quercetin have demonstrated synergistic reductions in the platelet adhesion, activation, and aggregation in vitro. Quercetin has demonstrated success inhibiting platelet aggregation. Single doses of quercetin glucoside, the naturally occurring form of quercetin, at 150 to 300 mg from food sources and high-quality dietary supplements was able to significantly inhibit the collagen-induced platelet aggregation in one small human study. However, long-term supplementation with 1 gram a day of quercetin aglycone, the form typically found in lower-quality dietary supplements, for 28 days had no significant effect on the platelet aggregation in healthy human volunteers. It should be noted the plasma concentrations of the quercetin in the former study were significantly higher than in the latter at the time of the aggregation measurements, suggesting the quercetin glycosides are absorbed more effectively than the quercetin aglycone. Quercetin from food sources like onions have had a positive trend on platelet aggregation. Salvia is a diverse genus of plants encompassing hundreds of species, many with ornamental, culinary, or medicinal importance. Salvia miltozoria, the red sage or danshen, is one of the most versatile Chinese herbal drugs. It's been used for hundreds of years in the treatment of cardiovascular disease, and still it's widely used as a standard thrombotic treatment in Chinese hospitals. Salvinolic acids A and B, the water-soluble polyphenols from the root, are responsible for its observed antiplatelet activity in animal models and in blood samples from healthy human volunteers. The seeds from Salvia hispanica, or chia, are rich in protein and the omega-3 fatty acid alpha-linolenic acid. In a small study on 27 patients with type 2 diabetes, the whole chia seed at 15 grams per 1,000 calories of food intake for 12 weeks showed significant reductions in the plasma fibrinogen and the platelet adhesion protein VWF. Small reductions in additional cardiovascular risk factors, systolic blood pressure, and HSC-reactive protein were also observed. Resveratrol has several effects on blood platelets, as was determined in vitro and in animal models, including the inhibition of platelet adhesion and aggregation, reduction in the secretion of clotting factors from the platelets, and inhibition of cyclooxygenase, the pro-inflammatory enzyme involved in platelet activation. Plasma resveratrol from consumption of red or white wine increases the release of nitric oxide from the platelets in healthy volunteers, inhibiting their activation. In an experimental study, the resveratrol was able to suppress the detrimental effects of homocysteine on the platelet aggregation and free radical generation. Grapeseed extract contains oligomeric procyanidins, and these support cardiovascular health through vasodilation and an increase in nitric oxide production. They've significantly reduced blood pressure in human trials. The grapeseed extract also exhibits antithrombotic activity in animals and in platelets isolated from healthy human volunteers. This might be related to the anti-inflammatory effect. In a small 8-week study of 17 postmenopausal women taking 400 mg of a flavonoid-rich grapeseed extract per day, a significant 23% lengthening of the clotting time compared to the control was observed on day 1 of the study. The increased clotting time indicates reduced platelet activation and aggregation. After 8 weeks, the difference in clotting time was not as significant, but it trended higher in the test group. Similar short-term reductions in platelet activity were also observed in a study on 23 male smokers. When combined with grape skin polyphenols, the grapeseed extract demonstrated better antiplatelet properties than either extract alone in animal models as well as on human platelets. Tomatoes contain several nutrients with established protective effects on the cardiovascular system. Lycopene has demonstrated hypotensive activity in humans, and several human trials indicate cholesterol-lowering effect. One mechanism by which the lycopene might limit the platelet aggregation is by activating cyclic GMP, a signaling molecule involved in vessel dilation. Tomatoes also exert potent antiplatelet activity in lab tests. The antithrombotic compound of the tomato are small molecules found within its water-soluble fractions. These are also high in soluble sugar content. Removal of the sugar increases the concentration of the tomato actives, and it stimulates their inhibition of the platelet aggregation by up to 50 times. 2 studies examine the effects of these standardized tomato extracts on platelet function in healthy human volunteers. High doses, which was 18 grams, or the equivalent to 6 whole tomatoes, and low dose, equivalent to 2 tomatoes, of the standardized tomato extract, both exhibited significant reductions in the platelet aggregation up to 6 hours after ingestion. Standardized bioactives from tomatoes suppress the platelet adhesion and aggregation by reversibly inhibiting P-selectin and 2 receptors necessary for clot formation. Pomegranates contain several bioactive antioxidant polyphenols, including the unique tannins punicalagins. The pomegranate juice consumption has been associated with significant decreases in blood pressure in hypertensive subjects and decreases in LDL cholesterol oxidation. Pomegranate juice polyphenols also function as vasodilators by supporting the endothelial function and as inhibitors of angiotensin converting enzyme, an enzyme that's associated with high blood pressure. 2 weeks of pomegranate juice consumption at 50 milliliters per day reduced platelet aggregation by 11% in a small study on 13 healthy individuals. In a human clinical trial, the pomegranate juice consumption was shown to prolong clotting time as little as 6 hours after consumption. Garlic's promotion of cardiovascular health has been substantiated by several human trials, particularly its blood pressure lowering activity and its ability to induce favourable blood lipid profiles. In cell models, garlic extracts inhibit platelet aggregation by reducing ion signalling that's involved in the platelet activation, and by increasing the synthesis of cGMP, a vasodilator. Garlic bioactives also promote endothelial nitric oxide release and enhance fibrinolysis. Moreover, the garlic inhibits the COX-1 and COX-2 enzymes, which suppresses TXA2 levels. The antithrombotic activity of garlic has also been the subject of several human trials in both healthy subjects and patients with cardiovascular disease, using aged extracts, water extracts, or garlic oil. The garlic has demonstrated reductions in platelet aggregation in each of these studies. Fish oil is a source of omega-3 fatty acids, EPA and DHA, and these are essential for several metabolic processes. Studies of tens of thousands of moderate and high-risk cardiovascular disease patients have demonstrated the ability of fish oil to reduce the plasma triglycerides, blood pressure, and the risk of cardiovascular mortality. Several human studies have observed the antithrombotic activity of fish oil, due in part to its ability to reduce the production of platelet aggregator thromboxane A2, a metabolite of the inflammatory omega-6 fatty acid arachidonic acid. Fish oil consumption decreases activation and aggregation and plasma fibrinogen levels. In type 2 diabetic patients, the pooled data from 3 human trials on 159 participants demonstrated a reduction in plasma fibrinogen by 32 mg per deciliter and platelet aggregation by more than 10%. The capsaicinoids, capsaicin and dihydrocapsaicin, are the major pungent constituents of chili peppers. The regular intake of chili peppers delays oxidation of serum lipids, and it lowers and improves insulin and glucose profiles following a meal, both of which contribute to reduced risk of cardiovascular disease. In animal models, capsaicin reduced platelet aggregation. An early study attributed the reduced plasma fibrinogen and the increased fibrinolytic activity in the native Thai individuals compared to the Americans living in Thailand to the amount of capsaicin in their diet. In lab tests, both the capsaicin and dihydrocapsaicin reduced the platelet aggregation and reduced the activity of clotting proteins in the blood sample from the 6 healthy patients. Ginger has been shown to inhibit platelet aggregation and to decrease the platelet thromboxane production in lab tests. Both raw and powdered preparations reduced the platelet aggregation in a small human trial. 5 grams a day of fresh ginger for 7 days inhibited the thromboxane production in 7 healthy volunteers, while 2 additional studies on a single dose of 2.5 grams of dried powder in 10 healthy volunteers and 10 grams a day of dried powder for 3 months in 30 patients with coronary artery disease also demonstrated an inhibition of the platelet aggregation. Doses lower than 2.5 grams had no effect in the human trials. Curcumin has a variety of protective roles in cardiovascular health, reducing oxidative stress, inflammation, and the proliferation of vascular smooth muscle cells and monocytes. Human trials revealed the effect of curcumin on reducing lipid peroxidation and plasma fibrinogen, both factors in the progression of atherosclerosis. Another mechanism by which the curcumin inhibits the platelet aggregation is by dampening the expression of P-selectin, an adhesion molecule that's expressed on both activated endothelial cells and platelets, and it mediates aggregation between the 2 types of cells. P-selectin also recruits leukocytes to the forming thrombus. In 8 subjects with abnormally high plasma fibrinogen, 20 mg of curcumin for 15 days reduced the fibrinogen level by nearly 50%. Experiments using human platelets or whole blood have demonstrated curcumin's ability to inhibit the platelet aggregation. French maritime pine bark extract, known as picogenol, has a strong anti-inflammatory and free radical scavenging effect, and it's been found to stabilize vascular collagen and prevent blood clots. In vitro research, an extract from New Zealand pine bark reduced cytokine-related expression of adhesion molecules by the endothelial cells, thereby reducing the likelihood of blood cell aggregation in response to inflammatory signalling. Pine bark extract can help travellers avoid blood clots during and after long flights. One study compared the effects of pine bark extract to placebo in 198 long-haul air travellers with a high risk of blood clots. The travellers received 200 milligrams of pine bark extract or placebo 2 to 3 hours before the flight, and 6 hours later, and 100 milligrams the next day. The flights averaged 8.25 hours. No thrombotic events occurred in those who were given the pine bark extract, but 5 occurred in the placebo group. In another trial, 186 travellers on a 7 to 8 hour flight received 2 tablets containing 150 milligrams of a proprietary blend of pine bark extract plus nattokinase, or placebo, 2 hours before travel and 6 hours later, and complete pre- and post-flight monitoring of edema and blood clots were performed. No thrombotic events occurred in the treated group, but 7 flight-related thrombotic events occurred in the placebo group. In addition, edema scores based on the measurements taken before treatment and after air travel decreased by 15% in the pine bark nattokinase combination group, and increased 12% in those receiving the placebo, with a significant difference between the two. One trial examined the effects of pine bark extracts on 156 participants who experienced a single episode of deep vein thrombosis. The participants were assigned to one of 3 treatments: pine bark extract, compression stockings, or both. After 1 year, 2 new episodes of deep vein thrombosis occurred in the participants using the compression stockings versus none in either of the groups receiving the pine bark extract. In addition, pine bark extract was more effective than compression stockings in reducing edema and as effective in improving microcirculation, while the combination of both was the most effective. This might be especially significant because compression stockings are historically associated with low compliance due to the discomfort. Findings from a clinical trial suggest the pine bark extract can help prevent thrombosis and other side effects in cancer patients who are undergoing chemotherapy and radiation therapy. The study included 46 cancer patients who began supplementing with 3 50 mg pine bark extract capsules or placebo after meals, for a total of 150 mg pine bark per day. After completing the first course of chemotherapy or radiation, after 2 months, the patients who received the pine bark extracts had decreased frequency of all investigated side effects, including thrombotic events, compared with those who were receiving the placebo. Fibrinogen, or factor 1, is a blood plasma protein and it's made in the liver. Fibrinogen is one of 13 coagulation factors that are responsible for normal blood clotting. When you start to bleed, your body initiates a process called the coagulation cascade, or the clotting cascade. Niacin, or nicotinic acid, or vitamin B3, is an essential nutrient with important effects throughout human metabolism. At doses substantially above the RDA, the niacin reduces the risk factors for cardiovascular disease and reduces the cardiovascular events and mortality. Some of the risk reduction is due to niacin's ability to significantly raise HDL cholesterol by up to 35% and reduce the amount of small, dense, low-density lipoprotein, or LDL particles, which are a risk factor for atherosclerosis. Niacin also lowers the plasma fibrinogen levels, a risk factor for cardiovascular disease. In the multi-center arterial disease multiple interventional trial, or ADMIT, the patients with peripheral arterial disease who were randomized to niacin, initially 100 mg per day, raised to 3,000 mg per day over the 12-month study, saw an average reduction of fibrinogen by 48 mg per deciliter, as well as a reduction in prothrombin time, a measure of blood clotting. Similar reductions in the plasma fibrinogen of 15% were observed in a 6-week study on men with elevated triglycerides. Vitamin C may possibly suppress fibrinogen levels, as is suggested by some association studies. A study involving more than 3,200 men in the UK found those with a higher plasma level of vitamin C also had lower levels of fibrinogen and superior endothelial function. Likewise, a study of 96 aging men and women found that an increase of dietary vitamin C of 60 mg per day, or the equivalent of about 1 orange, was associated with a reduction in fibrinogen that was estimated to cause a 10% reduction in the risk of ischemic heart disease. In an animal model, vitamin C was also shown to reduce the levels of VWF and fibrinogen. This suggests the inhibition of platelet adhesion and aggregation. Moreover, the vitamin C was able to reduce the blood pressure in this study. An experimental study found the incubation of fibrinogen molecules with vitamin C in vitro caused functional changes to fibrinogen. This might be associated with an impaired capacity for binding the surface of platelets. Fibrinolysis is a normal bodily process. It prevents blood clots that occur naturally from growing and causing problems. Primary fibrinolysis refers to the normal breakdown of clots. Secondary fibrinolysis is the breakdown of blood clots due to a medical disorder, a medicine or another cause. This can cause severe bleeding. Nattokinase is a fibrinolytic enzyme, an enzyme that breaks down the fibrin clots, and is found in natto, a soy fermented by the bacteria Bacillus subtilis. The bacteria produce the enzyme nattokinase, and it's not a metabolite of the soy. In lab tests, it reduces the platelet aggregation and the blood viscosity, and it enhances the fibrinolytic activity of the plasma in animal models. At a dose of 4,000 fibrinolysis units, or FUs, per day, the nattokinase has been shown to reduce the circulating fibrinogen and clotting factors, which are independent risk factors for cardiovascular disease, in patients who are undergoing dialysis or with cardiovascular disease, and in healthy volunteers. It was also able to reduce the frequency of deep vein thrombosis in 94 high-risk individuals on extended airline flights when combined with pine bark extract or pycogenol. The nattokinase has also been shown to reduce blood pressure in hypertensive individuals, and this might be attributed to its ability to lower the blood viscosity. Ethanol, or drinking alcohol in low doses, reduces the thrombotic risk by modifying the platelet function and reducing the platelet aggregation. As little as half a glass of red wine per day provides enough ethanol to reduce the thrombotic risk. However, higher doses of ethanol increase the risk of clotting substantially. All types of ethanol consumed in moderation, 2 drinks or less per day for men, and 1 drink or less per day for women, should reduce the thrombotic risk, but red wine also provides the beneficial polyphenols like quercetin and resveratrol. To learn more about blood clotting, check out my video on stroke recovery. To book a consultation or learn about herbs, supplements and natural treatments, check out my website. To learn more about blood clotting, check out my video on stroke recovery. To book a consultation or learn about herbs, supplements and natural treatments,